Hello everyone. In this video lecture, we are going to study about the associated digestive glands. The first we are going to see is the liver. The liver is the largest digestive gland. It is the largest exocrine digestive gland. Exocrine glands are those glands which have ducts to secrete their secretion. Endocrine glands do not have ducts for their secretion. It weighs almost 1.5 kgs. The main function of liver is to produce bile juice. Liver is present in the right side of the body in the abdominal region, and the liver does not have the function to produce any enzyme. The bile juice lacks enzyme. This is the only digestive juice which lacks enzyme. Rest all the digestive juices, be it saliva or the pancreatic juice, gastric juice, every juice has enzymes. But this particular digestive secretion lacks enzyme. So this is a particular thing about bile juice, that it does not have any enzyme in it. So let us now see the position of the liver and the different ducts. There are two major lobes in the liver. This is the right lobe and this is the left lobe. These are the two main lobes. There are two small lobes that are present and are not very distinctly seen. They are hidden at the back side. These are the quadrate and caudate lobes. These are small and these two are larger lobes. So in all, the liver has 4 lobes. From the left lobe, a duct arises. So we call it as the left hepatic duct, and this one from the right lobe. So this is the left hepatic duct and this is the right hepatic duct. Both of them fuse together to form a common hepatic duct. There is a gallbladder that is present. The gallbladder stores the excess of bile juice. Whatever bile juice is produced, the major function of the liver is to produce the bile juice. So the bile juice is stored in the gallbladder. There is another duct that arises. This is known as the cystic duct. This cystic duct combines with this common hepatic duct, and now this is called as the bile duct. This is the common bile duct or bile duct. In several of the books it's written as CBD, that is common bile duct, because it is the combination of all the 3 ducts. So this is the bile duct which actually brings the bile juice from the cells of the liver. And here is the stomach placed. Although we are not concerned with the structure of stomach, we have already seen, but to tell the position, this is the stomach and this ends up here. This is the temporary storage for the food, and from here the duodenum arises. Here is another gland, which is the pancreatic gland, the pancreas. It's a leafy structure and there is a pancreatic duct in the center. This bile duct arises from here, and then this duct from the pancreas joins with the bile duct to form a common hepatopancreatic duct. This is known as duct of Wirsung, named after the scientist, or pancreatic duct. The function of this duct is to bring the pancreatic juice which is produced by the pancreas, and then the pancreatic juice will come out through the pancreatic duct and will join with this common bile duct. The opening of this pancreatic duct into the common bile duct is by a sphincter. This sphincter is known as sphincter of Boyden. Sphincter of Boyden. All these are the names of the biologists, and this common hepatopancreatic duct, I'll write over here, this is the hepato. The hepato word is for the liver, pancreatic duct. Pancreatic duct for the pancreatic duct, that comes from the pancreas. Hepatopancreatic duct. Now this is one common duct, and this is bringing the bile juice from above, and this is the pancreatic juice from here, and they will open up into this portion of duodenum. Now when it opens up over here, it just forms this broad structure which is known as ampulla, and this structure, ampulla of Vater, all these things are so much asked in NEET, so we have to learn all these terms and sphincters. The common hepatopancreatic duct opens up into this rounded ball-like structure which is known as ampulla of Vater, which is guarded by the sphincter of OD, and this sphincter is sphincter of OD. So we will revise once again. This is the left lobe from where the left hepatic duct arises, and from here the right hepatic duct, and they form a common hepatic duct. This will fuse with the cystic duct which arises from the gallbladder. This is the gallbladder. The gallbladder is meant for the storage of bile juice. The cystic duct and the common hepatic duct fuse to form the common bile duct. The common bile duct then fuses with the pancreatic duct coming from the pancreas, and this juice will then come over here and will open up by the ampulla of Vater, which is guarded by the sphincter of OD. The opening of the pancreatic duct and the bile duct is guarded by sphincter of Boyden. So these are the two sphincters that are present over here. So this is how the pancreas and the liver are placed or located. Now inside the liver, if we take a section, if I take just a section, the cells are called as hepatocytes. How are they arranged? They are present in polygonal or hexagonal shape like this. So they are arranged in this manner. These are hepatocytes, the cells of the liver. It is the function of these hepatocytes to produce the bile juice, and they are rich in glycogen. It is said that the liver cells, that means glycogen is stored in liver, so where these cells, the hepatocytes, are rich in glycogen. Glycogen is stored in liver as well as in muscles of the body. So this is the reserve food material. Glycogen is the reserve food material which is stored in the liver. So this is how the cells, the hepatocytes, are arranged in the liver. So if we take an enlarged section, if I take this enlarged section of one hepatocyte, of one structure like this. This has an outer glissons capsule. This is fibrous outer structure which is known as glissons capsule, and in the center is the central vein present. Now actually the real cells which are present are inside, and they are all arranged as hepatic cords. So these are the hepatic cords, and around these hepatic cords are the hepatocytes arranged in this manner. So the real hepatocytes are these which are packed up in this structure. So these hepatocytes produces the bile juice. The bile juice is secreted in this small canal in between. The small canals in between are these. These small canals are called as canaliculi. So whatever bile juice is produced is actually then poured into the canaliculi. The canaliculi joins to form the Herring's duct in the center. The Herring's duct, which then opens into the bile duct that is present over here. So out of this, that means out of here there will be the bile duct that will be joining each hexagonal structure. So from here the bile duct will collect the bile juice. So the hepatocytes will produce it in the canaliculi. The canaliculi will pour it into the Herring's duct, and then it will be poured into the bile duct. One complete structure is known as hepatic cord, and all the hepatic cords are arranged in this plate like structure. So this is the arrangement of the cell. There are certain more cells that are present. There are certain fat cells that are present. These fat cells store cholesterol. So the liver also produces the cholesterol. These are adipocytes, fat storing cells in the form of cholesterol. When this becomes excess, then the extra cholesterol is released in the bile juice. And there are important cells which are present, which are irregular in shape. These are Kupffer cells. These remove all the toxic materials that are present. So these cells actually perform phagocytosis, and whatever toxin is present, they eat it up. So this liver also helps in detoxifying with the help of Kupffer cells. Now let us see the functions of liver. Liver is the busiest organ and it does multiple functions. So the first one, the major function is to produce bile juice. The bile juice has the pH which is between 8 to 8.5. That means this is alkaline. And this bile juice only helps in converting the acidic chyme into chyle. The chyme is converted into chyle in the intestine. Chyme is present in the stomach. This was the acidic food. We have already studied this. And chyle is present in the intestine. So we just now saw in this video that the bile juice is poured in the duodenum, which is the first part of small intestine. So here the food gets converted into chyle because of the high alkalinity. So 8 to 8.5 is the pH of the bile juice. Now what is the composition of bile juice? Bile juice contains water. This is for the reactions that are required, the metabolic reactions that are required, and for this the water is present. It contains bile salts. These bile salts only bring this pH, that is alkaline. The first is NaHCO3, which is sodium bicarbonate. Then the second one is sodium taurocholate, and sodium glycocholate. All these three are called as the bile salts. Then there are bile pigments. There are two bile pigments, bilirubin, this is yellow in color, and biliviridin, this is green in color. And both of them are actually produced by the red blood cells, the decomposition of the red blood cells after the 120 days when the RBCs are destroyed. So they are decomposed and then the bilirubin and biliviridin are formed. These are released in the bile juice because they are not at all useful, and this gives the color to the fecal matter. So whatever feces are produced, the color is because of the presence of the bilirubin and biliviridin. So these are the substances that are present in the bile juice. These are the regular substances present, and the excess of cholesterol and phospholipids are also produced in the bile juice. The liver is also involved in the metabolism of glucose, fat, and proteins. Let us see first the metabolism of glucose. Whatever glucose is consumed is converted into glycogen in the liver. This process is known as glycogenesis. The process of formation of glycogen is known as glycogenesis. So it is formed from the glucose. So glucose is converted into glycogen in the liver. The second metabolism of glucose is, this was the first one. The second one is that glycogen is also broken down when required. When there is improper intake of glucose, then the glycogen is broken down into glucose. This is by a process which is known as glycogenolysis. Lysis term is meant for breaking down. So glycogen is broken down in the liver and then the instant energy is provided by the glucose. The third is of metabolism of glucose is from the non-carbohydrate sources, that is from fats and proteins. Glucose is synthesized by the process called as gluconeogenesis. So what can be the non-carb sources? It can be either amino acids, proteins, and fats. Glucose is so important for the body. So when there is no carbohydrate intake, from the amino acids, proteins and fats also, the glucose can be synthesized by the process of gluconeogenesis. This also takes place in the liver. So the three processes are involved in the metabolism of glucose. Now for the metabolism of fat. The fat is synthesized from fatty acids and glycerol to form the fat. So when fat is formed, the process is known as lipogenesis. This process takes place in the liver. And when fat is again broken down to form the fatty acids and glycerol, it is known as lipolysis. So these two processes are involved in the metabolism of fat. Now let us see the metabolism of protein. In metabolism of protein, there is transamination. Transamination is the process in which one amino acid is transferred to another protein. So the position trans means to shift. So transamination. There is another one which is known as deamination, which is the removal of an amino acid. So these two are the processes. Then there is another function of the liver. Let me mark it as 3. We were studying the second function, the metabolism of glucose, fat and protein. And now let us see the third function. And in the liver, the harmful ammonia, which is formed when the transamination, deamination and metabolism of protein is being done, at that time ammonia is formed. Ammonia is very toxic and we cannot release ammonia from our body, and so it is converted into a less harmful substance which is called as urea in the liver. So ammonia is converted into urea in the liver by a process which is known as ornithine cycle. This has one more name by the name of the scientist Krebs-Hasselbalch cycle. Do not confuse it with Krebs cycle, which is a part of respiration. Krebs-Hasselbalch cycle is the ornithine cycle, which is by the conversion of ammonia into urea. So the production of urea is in the liver, but the elimination or the removal of urea is by the kidney. Then the fourth function is the storage of fat soluble vitamins. These are 4: vitamin A, vitamin D, vitamin E and vitamin K. So these are fat soluble vitamins. These are also stored in the liver. Then the liver helps in thermoregulation. It helps in proper distribution of heat. The liver also helps in detoxification. Whatever toxic substances are present, it removes it with the help of the Kupffer cells. So we have seen the major functions of the liver. So this is all about this digestive gland, liver, which is the largest digestive gland. If you have any doubt, you may ask me in the comment section, and you can also refer to the questions that are given in the description box. Thank you.